Hey guys, it's Max. I'm going to teach you guys about the two experiments that helped us figure out the genetic code. Now I started with two guys, Nuremberg and Mate. They were able to figure out 3 codons and the amino acids they coded for each. So how did they do that? Well, they started with 2 things, a poly-U strand and a cell-free extract. The poly-U strand was basically just the whole string of U's of uracils, and the cell-free extract was a cell that had been basically gutted. Nothing was in it. And they took those 2 things and combined it into 20 different bottles. In each of those bottles, they added an amino acid, a different one. And they found that the only amino acid that actually responded to those poly-U strand was phenylalanine. So they were able to figure out that the codon UUU coded for phenylalanine. So they did that experiment 3 more times with a poly-C strand, a poly-A strand, and a poly-G strand. And so they were able to figure out that the poly-C strand coded for proline, the poly-A strand coded for lysine, and the poly-G strand actually wasn't able to code for anything, so they had nothing. And so that was a good start, but however, there were about 64 codons, so we had a lot more work to do, and they only had 3. So they had to do another experiment. This time it was at Nuremberg, and he picked up another guy, Liter. And so they were able to identify the remaining 61 codons and their respective amino acids. How did they do that? So they were able to isolate every single combination of codon into a short little mRNA strand. And they took that little mRNA and they basically did the same thing as with the other experiment. They did each codon was used 20 times. So they took each codon and they combined it with a ribosome and a tRNA with a different type of amino acid with it. And they did that for every single codon combination. So it was a lot of trials, but they were able to figure out every single amino acid and what codon coded for that amino acid. So they were able to find that CUA coded for leucine, AGU coded for serine, GGC coded for glycine, GUU coded for valine, and then there were 57 other codons that binded with their respective amino acids. Basically, they were able to find out every other codon that hadn't been figured out from that previous experiment. They were able to isolate that and figure out what amino acid coded for that. So there you have it. In these 2 experiments, we were able to figure out the entire genetic code and what amino acids coded for what codon. Thank you all.